She did everything the way she'd been told. Whole grain toast, a banana, black coffee, out the door by 7:15. And by 10:30 most mornings, she was staring at her second cup of coffee, wondering why her hands felt slightly shaky, why the fog behind her eyes had rolled back in less than 3 hours after she'd eaten what she genuinely believed was a responsible breakfast. She started keeping a note on her phone. Ate at 6:50, crashed by 10:15, same the next day, and the day after. She began to suspect something was wrong with her, some flaw in her metabolism nobody had diagnosed, some willpower she was quietly failing to summon. What she didn't know, what almost nobody tells you, is that her body was running 2 separate clocks that morning, and only one of them had gotten the memo that the day had started. Somewhere behind your eyes, in a cluster of roughly 20,000 neurons called the suprachiasmatic nucleus, sits the master clock of your entire body. It takes its orders almost exclusively from light. The moment sunlight, or a bright enough approximation of it, hits your retina, that cluster of neurons updates its internal sense of time and starts broadcasting the news outward. It is morning. The active phase has begun. Get ready. Her eyes had received that broadcast the second she opened the blinds. Her gut never did. Here is the detail almost nobody knows, the one that this entire video is built around. Your liver never receives that broadcast directly. Neither does your pancreas. Neither does the lining of your intestine. Every one of those organs runs its own separate clock, a loop of genes switching each other on and off across roughly 24 hours, discovered in enough molecular detail to win Jeffrey Hall, Michael Rosbash, and Michael Young the 2017 Nobel Prize in Physiology or Medicine. These are called peripheral clocks, and they take their primary cue not from light, but from food, and not from just any food. From a specific narrow category of it. The genes involved have names that show up throughout circadian research: clock, BMAL1, PER1 and PER2, CRY1 and CRY2, a set of proteins that activate and repress each other's expression in a loop that takes almost exactly a day to complete one full cycle. Every cell in your liver, your pancreas, and your intestinal lining runs a version of this same molecular loop, and under normal conditions it needs regular resetting to stay accurate, the way a mechanical clock drifts slightly and needs occasional correction against a more reliable reference. For your brain's master clock, that reference is light. For the loop running in your liver cells, the reference is the arrival of nutrients, specifically at a time consistent enough, and specifically of a kind the loop actually recognizes, that it can calibrate against them. This is the entire crux of the matter, and it is the answer to what was actually happening to her. Light sets the clock in your head. Food sets the clocks in your gut, but only certain food actually counts as a legible signal to those clocks. Toast and a banana are calories. They are not, in the specific molecular sense this script is about to walk through, information. When you wake up to morning light, and your gut receives nothing that qualifies as a real timing signal, the two systems disagree. The conductor in your brain is keeping one tempo, and half the orchestra in your abdomen is still playing to yesterday's rhythm. The medical term for this disagreement is circadian misalignment, and it turns out to be one of the quieter drivers of the exact mid-morning crash she had started writing down on her phone, convinced it was a personal failing, rather than a body running 2 clocks that had never been properly introduced to each other. The obvious next question is what actually counts as a real signal? Not any food, not any calorie. The gut is listening for something more specific than that, and out of the entire universe of things you could eat for breakfast, the research points to exactly 3 categories of food that reliably deliver the message your peripheral clocks are waiting for. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. Before any of those 3 foods enter the conversation, it helps to understand what state your gut is actually in when you open your eyes, because it is not a blank slate waiting for instructions. It is mid-task. While you slept, a slow contraction began in your stomach roughly every 90 minutes, and traveled the entire 20-foot length of your small intestine, sweeping downward like a maintenance crew working an empty building after closing time. Physiologists call this the migrating motor complex, and it runs only when the gut is empty, clearing out the debris left from yesterday's meals. The fiber your enzymes couldn't fully break down, cells shed from the intestinal lining, bacteria that wandered somewhere they shouldn't be. That low growl an empty stomach makes has less to do with hunger than most people assume, and more to do with this overnight cleaning cycle finishing its final pass. At roughly the same time, in the hour before you naturally surface from sleep, your adrenal glands begin releasing cortisol in a sharp rising tide, a rhythm called the cortisol awakening response, one of the most reliable hormonal shifts in the entire 24-hour cycle. It occurs in the overwhelming majority of healthy adults regardless of how well they slept or what day of the week it is, climbing to its highest point roughly 30 to 45 minutes after you open your eyes. Cortisol's job here has nothing to do with stress in the way most people picture it. It tells your liver to release stored glucose into the bloodstream so your brain and muscles have fuel to move before you have eaten a single bite. So consider the state your body is actually in during those first waking minutes. Cortisol is high and climbing. Your liver is releasing glucose on its own, without any food involved at all. Your gut has just finished, or is finishing, its overnight sweep. And your insulin sensitivity, the responsiveness of your cells to the hormone that lets glucose enter them, sits at one of its highest points of the entire day. None of this is random. It is a system that evolved to expect food shortly after waking and spent the entire night preparing for its arrival. The table has already been set. There is even a preparatory step that happens before the first bite is fully swallowed. The sight, smell, and anticipation of food trigger what physiologists call the cephalic phase of digestion, a signal traveling down the vagus nerve from the brain to the stomach, pancreas, and gallbladder, telling the stomach to begin releasing acid, the pancreas to prepare digestive enzymes, and the gallbladder to ready its stored bile, all before a single molecule of food has actually reached the small intestine. The digestive system does not wait passively for the meal to arrive. It starts working in anticipation, the way a kitchen begins firing up the moment an order is called out, rather than waiting for the plate to be kneaded. The only open question is what, specifically, gets placed on that table, and whether that specific thing is capable of telling every peripheral clock in your body that the meal it has been waiting for has finally shown up. The first food category that qualifies as a genuine morning signal is soluble, fermentable fiber, and the reason it counts as a signal, rather than just a nutrient, traces back to an experiment that had nothing to do with breakfast at all. Researchers studying circadian biology in mice found that when the animals ate on a schedule aligned with their normal active period, their gut bacteria oscillated in a clean, predictable daily rhythm, specific bacterial populations blooming at consistent hours and receding at others. When the feeding schedule was deliberately scrambled, food arriving at times that conflicted with the animal's internal clock, those bacterial rhythms flattened out. The same species were still present. Their daily choreography simply collapsed, and the consequences reached well beyond the gut itself, altering how the animals processed fat and sugar and nudging them toward weight gain and glucose intolerance. The bacteria were keeping time, and when their clock lost its reference point, the disruption rippled outward into the host's entire metabolism. Fermentable fiber is the primary fuel that entrains that rhythm. Your own digestive enzymes cannot break it down, but the bacteria living in your large intestine can, and when they ferment it, they produce a class of molecules called short-chain fatty acids, chiefly butyrate, propionate, and acetate. Butyrate is not a byproduct to be tolerated. It is the preferred fuel source for the cells lining your colon, the energy they burn to maintain the wall separating the contents of your gut from your bloodstream. A well-fed bacterial population producing abundant butyrate keeps that wall strong and sealed. A population that goes unfed, or fed on the wrong schedule, produces less of it, and the wall begins to weaken, allowing fragments of bacterial membrane to leak into circulation, where the immune system reads them as a low-grade chronic threat. Whole rolled oats or steel-cut oats sit at the center of this category for a specific, well-documented reason: their beta-glucan content. The beta-glucan fiber in oats has been studied more extensively than almost any other single dietary fiber. thoroughly enough that the FDA approved a heart health claim for it back in 1997, based on evidence that it reduces LDL cholesterol through bile acid sequestration, moderates the blood sugar response after a meal, and specifically feeds populations of lactobacillus and bifidobacterium, two of the most studied beneficial bacterial genera in the human gut. Adding chia seeds, which contribute both soluble fiber and omega-3 fatty acids, or flaxseed, rich in lignans and additional soluble fiber, extends the same substrate further, giving a broader range of bacterial species something to ferment in that critical early window. The distinction worth holding on to here is between fiber that merely passes through and fiber that actually gets fermented and used as a timing cue. Highly processed cereal grains, stripped of much of their original fiber architecture during manufacturing, provide comparatively little of this signal, even when the box advertises whole grains prominently. The fiber has to still be structurally intact enough for bacteria to do something with it. Oats, largely unprocessed, remain one of the most reliable sources of a signal that is genuinely doing the entraining work, rather than simply adding bulk. There is a further reason a well-timed fiber signal matters beyond gut wall integrity and blood sugar. The overwhelming majority of the body's serotonin, more than 90% by most estimates, is produced not in the brain, but in the gut, manufactured by specialized cells called enterochromaffin cells, that line the intestinal wall like sensors embedded along a pipeline, tasting what passes through and responding to the specific chemical signals bacteria produce during fermentation. Short-chain fatty acids generated from a well-fed, well-timed fiber intake influence how actively these cells produce serotonin, which in turn shapes the signal traveling upward through the vagus nerve toward the brain. A morning fiber signal does not stay confined to the gut. It participates hours later in a chemical conversation that reaches as far as mood and mental clarity, a connection that has nothing to do with wellness marketing and everything to do with a nerve carrying real, continuous, chemically shaped information from one end of the body to the other. The second category is protein, and the reason it counts as a distinct morning signal rather than simply fuel comes down to a specific window of heightened receptivity in muscle tissue itself. Muscle protein synthesis, the ongoing process by which your body builds and maintains muscle, does not run at a constant rate around the clock. It runs in cycles, and the morning window following an overnight fast is one of the periods when muscle tissue is most primed to take up incoming amino acids and put them to structural use. Research on protein distribution across the day, including work from Douglas Padden Jones at the University of Texas Medical Branch, has consistently found that spreading protein intake across 3 meals, breakfast included, produces meaningfully greater 24-hour muscle protein synthesis than concentrating the same total amount into 1 or 2 larger meals later in the day. The common modern pattern, close to no protein at breakfast, followed by an oversized serving at dinner, wastes a meaningful share of that protein's potential, because the body appears to have a functional ceiling for how much of a single dose it can put toward active synthesis at once, somewhere in the range of 25 to 40 grams per meal. Eggs sit at the center of this category for reasons that go beyond convenience or tradition. 2 eggs deliver roughly 12 to 14 grams of complete protein, containing all 9 essential amino acids the body cannot manufacture on its own. They are also one of the richest common dietary sources of choline, a nutrient critical for liver function and cell membrane integrity that a majority of adults in Western diets fall short on without particularly noticing. And they carry a meaningful dose of leucine, the specific amino acid most directly responsible for triggering the cellular signaling pathway that switches on muscle protein synthesis in the first place, present in amounts that approach the threshold research has identified as effective for triggering that response. Pairing eggs with Greek yogurt or cottage cheese, both concentrated sources of the slow-digesting protein casein alongside faster-digesting whey, is enough to comfortably reach the 25 to 40 gram range in a single sitting. There is a second layer to why protein functions as a timing signal, specifically in the morning, rather than simply being good nutrition at any hour. Insulin sensitivity, the efficiency with which your cells respond to rising blood sugar, follows its own daily rhythm, governed by the same peripheral clock system discussed earlier, and it peaks in the morning before declining through the day. Pancreatic beta cells are primed and responsive early on, releasing insulin briskly, and cells receive that insulin efficiently. The identical meal eaten in the evening, when that same insulin response has grown comparatively sluggish, produces a larger and more prolonged blood sugar excursion, a difference that has been measured directly in controlled research settings, including work out of Frank Shear's laboratory at Harvard Medical School examining the metabolic consequences of circadian misalignment. The morning meal's influence does not end when the meal itself is over. A phenomenon called the second meal effect, first described by David Jenkins at the University of Toronto, the same researcher who developed the glycemic index, found that the fiber and protein composition of breakfast measurably changes how the body handles carbohydrates at the very next meal, hours later. Soluble fiber consumed at breakfast slows gastric emptying and produces a more gradual absorption curve that appears to extend its influence into lunch, blunting the blood sugar response to a meal eaten well after the first one has already been digested. The choices made in that first hour are not isolated to that first hour. They set a metabolic tone that persists into the middle of the day, which is part of why the specific composition of breakfast carries more downstream weight than its modest share of daily calories would otherwise suggest. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. The third category is fermented food, and it earns its place here on the strength of a specific, fairly recent clinical trial rather than tradition or folklore. In 2021, researchers at Stanford, including Hannah Waystick, Gabriela Fragiadakis, and senior authors Justin and Erica Sonnenberg, alongside Christopher Gardner, published a controlled study in the journal Cell, comparing two dietary strategies over 10 weeks in 36 healthy adults. One group increased their intake of high-fiber foods. The other increased their intake of fermented foods, specifically yogurt, kefir, fermented cottage cheese, kimchi, sauerkraut, and kombucha. The fermented food group showed a measurable increase in overall microbiome diversity, with the effect scaling alongside the size of the servings consumed, along with a documented decrease across 19 separate inflammatory markers in their blood, and reduced activation in 4 types of immune cells. The high-fiber group did not show the same diversification over the same window, and participants in that group who started with lower baseline microbiome diversity actually showed increases in some inflammatory markers rather than decreases. The researchers were explicit that these findings were preliminary and that the underlying mechanisms need further study, but the directional signal was clear enough to reshape how a number of researchers in the field talk about fermented food specifically, as distinct from fiber generally. Why fermented food functions as a timing signal in particular, rather than simply a health food to eat at some point in the day, comes back to the same peripheral clock logic running through the other two categories. A serving of live culture yogurt or kefir delivers a population of active microorganisms directly into a gut that has just finished its overnight housekeeping sweep and is entering its most metabolically receptive window of the day. That population arrives alongside protein, in yogurt's case, and alongside its own fermentation substrate, meaning the delivery is not just organisms, but organisms bundled with something to immediately do. Plain Greek yogurt is the most accessible entry point into this category, functioning simultaneously as a protein source and a fermented food source in a single serving, which is part of why it shows up repeatedly across morning nutrition research as an efficient choice rather than merely a popular one. It is worth being precise about what fermented food is not doing here, because the claims in this space tend to drift toward the vague. This is not a detox mechanism, and nothing about fermented food is flushing anything out of your system in the sense that marketing language around the word implies. What the research actually supports is narrower and more interesting, a measurable shift in the diversity and behavior of an existing bacterial population, and a documented reduction in circulating inflammatory markers over a period of weeks. That is a real, specific, replicated finding. not a vague wellness claim borrowing the language of biology without its substance. The specific bacterial residents worth knowing by name make the case more concrete than a general reference to microbiome diversity. Akkermansia mucinifila, a species that has drawn substantial research attention over the past decade, lives specifically within the mucus layer of the gut and feeds on mucus components as its energy source. An arrangement that sounds like it should erode the lining, but appears to do the opposite, since the bacterium's activity stimulates the gut to continuously produce fresh mucus in response, functioning less like an erosive presence and more like a maintenance crew that triggers renovation simply by being active. Research associated with Patrice Cani's laboratory in Belgium has linked higher Akkermansia abundance with lower rates of metabolic syndrome, insulin resistance, and obesity. Faecalibacterium prausnitzii, one of the most abundant species in a healthy human gut and one of the primary producers of butyrate discussed earlier, is consistently found at sharply reduced levels in people with inflammatory bowel conditions, a pattern researchers increasingly treat as structural rather than incidental. A well-timed fermented food signal is, among other things, an opportunity to support the specific populations these named species represent, not simply an abstract gesture toward gut health in general. Understanding what counts as a signal is only half the picture. The other half is understanding why so much of what fills an ordinary modern breakfast plate, despite arriving at the right hour, fails to register as one. Start with orange juice, because it illustrates the principle cleanly. A standard 240 milliliter glass of commercial orange juice contains somewhere between 21 and 26 grams of sugar, comparable to a can of soda, with virtually none of the original fruit's fiber intact, since that fiber was removed during extraction. On an empty stomach, with nothing to slow its absorption, that sugar load hits the bloodstream quickly, provoking a sharp insulin response followed, in many people, by a drop in blood sugar below baseline a couple of hours later, a pattern called reactive hypoglycemia, and a leading, underappreciated cause of the mid-morning energy crash usually blamed on inadequate sleep. A whole orange, fiber intact, carries a glycemic index somewhere around 35 to 45. The juiced version, stripped of that fiber matrix, sits closer to 50 to 65. The fiber was never incidental to the fruit. It was the mechanism controlling how the sugar arrived. Sweetened breakfast cereal follows a similar pattern at a larger scale. Most commercial varieties carry somewhere between 10 and 25 grams of added sugar per cup, alongside 1 to 3 grams of protein, and the grain fiber that remains after processing has typically been broken down enough during manufacturing that its usefulness as bacterial fuel is minimal. The result is a food that spikes blood sugar, offers muscle tissue almost nothing to work with during its most receptive window, and preferentially feeds sugar-loving bacterial populations rather than the fiber-fermenting species that produce butyrate. None of this reflects a poor decision made deliberately. It reflects a cultural default assembled long before this particular biology was well understood. Coffee deserves separate treatment because the mechanism involved is more specific than simple acidity. Caffeine directly stimulates cortisol release, and drinking coffee within that first 30 to 45-minute window, precisely when the cortisol awakening response is already climbing on its own, stacks a second cortisol stimulus on top of the first. Cortisol, held elevated for too long, suppresses the protective mucus layer lining the stomach wall, the buffer standing between that layer and gastric acid sitting at a pH between 1.5 and 3.5, an acidity level in the same range as battery acid diluted. Andrew Huberman's public research communications have pointed to 90 to 120 minutes after waking as an ideal caffeine delay, specifically to avoid this stacking effect, though a shorter delay of even 30 to 45 minutes meaningfully reduces the overlap, even when the longer window isn't realistic on a given morning. None of this makes coffee itself the problem. It makes the timing, and specifically the presence or absence of food alongside it, the actual variable worth adjusting, since drinking coffee alongside a meal, rather than before one, changes how its compounds interact with an already buffered stomach lining. Warm water belongs in this discussion too, not as a fourth signal food, but as useful preparation for the three that matter. A study published in the World Journal of Gastroenterology found that warm water specifically, compared to cold, enhanced gastric motility and improved measured gastric emptying rates, likely because warm fluid relaxes digestive smooth muscle in a way cold fluid does not, while cold water can trigger a mild, temporary constriction of blood flow to digestive tissue. A glass or two of warm water before anything else rehydrates tissue that has gone 7 or 8 hours without fluid, stimulates fresh mucus production, and activates peristalsis, effectively warming up the system before the three real signals arrive, rather than delivering a timing signal of its own. Underlying all of this is a piece of anatomy most people never think about. The gut runs its own semi-independent nervous system, sometimes called the enteric nervous system, containing somewhere between 200 and 500 million neurons woven through the wall of the digestive tract, a number comparable to the nervous system of an entire cat. Michael Gershon at Columbia University spent much of his career documenting this network in detail, giving it the popular name the second brain. That network does not sit passively waiting for instructions from the skull. It coordinates digestion largely on its own, and continuously reports its status upward through the vagus nerve, which means the specific composition of a meal and the specific hour it arrives is not simply information the stomach processes in isolation. It becomes part of a running conversation between two nervous systems, one of which happens to be the one generating conscious thought. None of this argues for a rigid, one-size-fits-all ritual, and the honest caveats matter as much as the mechanisms themselves. The 30-minute window discussed throughout this script is not a hard threshold where biology flips from harm to benefit at the 61st second. The underlying reality is a gradient, not a switch. Some people wake with no appetite at all, and forcing food into an unwilling gut creates its own kind of disruption rather than solving one. Some people function well on intermittent fasting patterns that deliberately delay the first meal by hours, and for them, the same three-signal logic still applies whenever that first meal does arrive, since the alignment that matters is between food timing and the body's broader light and activity signals, not a specific number of minutes since waking. With that said, for most people living an ordinary schedule of morning light and daytime activity, a coherent sequence looks something like this: warm water first, before coffee, before food, before checking a phone, taking advantage of the gastric motility research without asking it to do more than it actually does. A deliberate pause of 30 to 45 minutes, longer if practical, before coffee, letting the cortisol awakening complete its natural arc, without a second stimulus layered on top of it. Then, within roughly 1 to 2 hours of waking, a meal built specifically around the three categories this script has walked through: a genuine fermentable fiber source, like whole oats, ideally with chia or flax added, an adequate protein serving in the 25 to 40 gram range, with eggs as an efficient anchor, and a fermented food component, plain Greek yogurt being the single most efficient way to fold that signal into the same meal that is already delivering protein. The response to getting this right is faster than most people expect. Measurable shifts in microbiome composition have been documented in research settings within 3 to 4 days of a consistent dietary change, not weeks or months. The gut is not keeping score of years of mistimed breakfasts and waiting to be paid back before it starts cooperating. It responds to what arrives now, on the schedule it receives it. None of this requires expensive ingredients or a specialized shopping list. Oats, eggs, and plain yogurt are among the least expensive whole foods available in most grocery stores, which makes the gap between what the research supports and what most modern breakfasts actually contain less a matter of access and more a matter of information that never made it from a physiology journal into an ordinary kitchen. She eventually swapped the toast and banana for oats with a spoonful of yogurt stirred in and 2 eggs on the side, still black coffee, just later. The note on her phone stopped getting new entries within the first week, not because she was trying harder, but because the 2 clocks running her morning had finally been given the same information at the same time. Tomorrow morning, before any of it happens consciously, the cortisol will already be rising, the liver will already be releasing glucose, The overnight sweep through the small intestine will already be finishing its last pass, and every peripheral clock in your digestive system will be waiting for exactly one thing, a specific, recognizable signal that the active phase of the day has genuinely begun. Only 3 categories of food reliably send it. Everything else on the plate is just calories arriving at the wrong department, on time for nothing in particular. Thank you so much for watching until the end. If you loved this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.